Good afternoon. Welcome to the 8th Centennial Lunch and Learn lecture. Today's lecture is called The Glass Menagerie, Animals' Central Role in the History of the School of Public Health. I'm Karen Thomas. I'm the Bloomberg School historian, and this talk is based on research that I did for my book that will be coming out on the history of the school with Johns Hopkins University Press. It is called Health and Humanity, and it will be out in June, and I'm very excited about it. So my talks have approached the history of the school from a number of different directions, but this one is fairly unique in using animals to frame the talk. And I'm going to jump around quite a bit chronologically, but I'm going to try to walk us through some major themes. Understanding why animals were just absolutely essential from the founding of the school forward, and to ask what have we learned from our research on animals, and also asking about how and why the priorities and methods of animal research changed over time. And then finally, looking at how the fields of global health and environmental health have really transformed the place of animals within our public health ethical framework. I'm going to go fairly rapidly, and there's a lot of information on the slides, but if you can hold your questions to the end, I can always go back. So, in my talk on the dynamic science of public health, I emphasized that William Welch was first and foremost a basic scientist, and he used animals extensively in his own laboratory research. And both Welch and William Osler, two of the four founding fathers of the Johns Hopkins Medical School, were great advocates of comparative pathology, that is, using animal models to study and better understand human health. And in fact, when I was doing my policy lecture research and looked at what Welch was testifying before Congress about, one of the things he really got very riled up about was the anti-vivisectionist movement, as it was called in the late 19th and early 20th centuries. So the anti-vivisectionists were the PETA of today. So it may seem obvious to us now, but public health research focused on animals in part because a lot of public health problems, especially in infectious disease, came from increased human-animal contact. So in rural, coastal, and tropical areas, and I grew up in the Gulf Coast of Florida, which was all three, humans are in much more contact with both domestic and wild animals through agriculture, through fishing, timber, things like that, different kinds of occupations place people in close contact with animals, and especially in developing countries, and it used to be the case in this country, people actually live with their animals. Certainly, agriculture presents the opportunity for both toxic and infectious exposures between human and animals. And more recently, we have started to look at the effects of factory farming on animal and human health as another aspect of this human-animal connection. One of the things that William Welch was most famous for that seems really weird to me now, but it made a lot of sense then, was he did detective work to solve an epidemic of hog cholera in Maryland. And more than his internationally famous other research, that hog cholera epidemic sealed his fate in Maryland. I mean, people just absolutely loved him for it and for saving their hogs. And people were still writing to him decades later, after he became dean of the School of Hygiene, asking them to come help figure out what was wrong with their animals. And in fact, one guy wrote from South Georgia and said, all our hunting dogs are dying and we don't know why. Can you send someone down to help us out? And by this time it was the late 1930s, and they said, we're sorry, we don't really do that anymore, and referred them to the US Department of Agriculture. But that just gives you a sense that even William Welch was like right in the thick of this animal-human interaction. There's a lot, I had to learn a lot about why different animals are specifically chosen for different types of research, and I think it's worth just mentioning quickly. There's very practical considerations, cost and how difficult they are to work with or maintain, the size of the animal, the lifespan of the animal. Richard Markham here in the picture was going to work with camels because he was trying to produce a type of bacteria that would help him to produce an antibody to AIDS. But the camels were too mean, and the llamas were also too mean. Llamas are actually, I found out, used as guard animals for like herds of sheep. So he went with the alpacas because they were much nicer. So if you're doing chronic disease research, you want to use something like sheep because you can watch the progression of disease over a longer lifespan. Whereas rodents are almost the universal choice for many types of genetic research because, as we all know, they reproduce very rapidly and you can see changes in generations much more easily. And asthma research is certainly a major emphasis for our school. And Dr. Mitzner told me that rodents don't breathe through their mouth or cough or wheeze, so they're no good for asthma research, but other animals do. So some of the purposes of animal research. So these photos are courtesy of Dr. Mitzner. So the photo on the left is of the different models of animal lungs that he uses in his research on lung physiology. And then there's also a perfused pig lung, which basically the organ and the tissue is kept alive by pumping blood through it. So the anatomy and physiology of these animals' systems are matched with specific disease types. So the pig lung and pig hearts also are frequently used in heart and lung research because they can tell us, there are a lot of similarities to human anatomy, and they can tell us a lot about what we want to know about disease in those systems. But animal models are very, very useful to do research on either infectious and or parasitic organisms. We can basically learn by observing, by exposing an animal to a disease and seeing how it reacts. But we can also, some pathogens, such as malaria, can't be cultured outside a human being. And therefore, by using a model of bird malaria, the type of malaria that affects birds, we can actually experiment and learn a lot about how malaria is transmitted and how we might be able to stop it. So there's all sorts of zoonotic disease that has animal vectors, rabies, typhus, malaria, hantavirus, salmonella, which is mainly spread through reptiles, and on and on. And it's important to note that some diseases act one way in urban areas where there's mainly humans around and another way in rural or jungle areas. So that, for instance, with typhus, it's spread by arthropods such as lice, but it stays in a mammal reservoir, and then when humans come in contact with the animals, it's transmitted to humans. I can't emphasize enough the importance that field biology has had to our school. Kenneth Maxey, one of our very first graduates, and you're actually going to see a picture of him in the next Public Health magazine, he became chair of epidemiology after his mentor, Wade Hampton Frost, and he spent the years that he was a young man as a federal surveyor with the U.S. Forestry Service and Bureau of Indian Affairs. And he went out in the American West and he camped and he mountain climbed and he just was out in the environment. And this generation of people who really founded our school and were on the early faculty, they lived at a time when America was overwhelmingly rural, when you really had to have a lot of wilderness survival skills still, and that it was just a very natural thing for kids. And I mean, I know growing up as a kid in Florida, I went out by myself when I was a very little kid and like got in the bayou and went fishing and all that. And that's a key part of what shapes these faculty members' understanding of the world around them and of public health. I think I love the quote that the way Carl Taylor opened his medical school essay to apply to Harvard. My study of anatomy began with dissecting a tiger to see where the food went. Of course, who wouldn't? So I'm showing several examples. You know, there's Carl Taylor, absolutely up to almost his armpits, you know, in the water out in the Indian countryside. Kirti Shaw's work with rhesus monkeys in India was an essential part of his event. He first worked with SV40, simian virus, in monkeys, and he was trying to figure out if SV40 transmitted cancer to humans. And he found out after about 40 years that it did not. But his experience with that type of virus helped him to very deeply understand HPV, which was a similar virus type, and that does, of course, cause cancer. It's the main cause of cervical cancer. So being out in that environment and watching the interactions of people, animals, plant life, all of that was just a central part of the experience here. I'm not going to talk much about entomology, but one of the greatest entomologists of the 20th century was Lloyd Roseboom, who was both an alum and he served on the faculty for decades. And he literally wrote the book on identifying malarial mosquito species. And that knowledge saved a lot of soldiers' lives during World War II. And there he is in his military uniform. So I had mentioned the different animal models. So I'm going to go through a couple of different animal models and look at how they were matched with different diseases. So I saw on the Centennial slideshow on the Wall of Wonder that it mentions Robert Hegner is really one of the main founders of the discipline of medical zoology. So he was chair of medical zoology and I can't remember the full name. It was a long scientific name of his department, and it was one of the predecessors of what's now microbiology and molecular immunology. But his lab, with his intense knowledge of both malaria parasites and insect species and birds, he is the one that really helped to find the best match of the bird malaria and the right bird species to infect so that he could study this interaction of the host and agent. He used to use canaries from Baltimore pet stores. The pretty yellow ones were the really popular ones, but the poor males weren't so good sellers, so they ended up in Robert Hegner's lab. His student, Ernest Hartman, developed a specific type of bird malaria model that was one of the most productive models for malaria research because it behaved in a lot of erratic ways that mimicked human malaria. So during World War II, the sixth floor of what is the main original hygiene building was filled with ducklings. And what they were doing with those poor little ducklings is that Peking ducklings of all bird species were particularly susceptible to malaria, so it was very easy to inoculate them. And they were chosen to host the, I'm sure I'm not going to pronounce this right, the P-Lofere model. So understanding structure-activity relationships from an immunological perspective is absolutely essential to developing an effective therapy for malaria. And that's exactly what they were doing. The entire National Research Council Office for the Survey of Anti-Malarial Drugs was based here at Johns Hopkins. And a faculty in the medical school headed it, and I'm sorry, his name is escaping me right now, but that research office, it coordinated all the other research offices around the country, but it also tested over 14,000 drugs, so that's more than 10 a day for 4 years throughout the war. So that's what all those ducklings were doing on our sixth floor. And out of all of those different possible combinations of drugs, they identified a drug called chloroquine, which was so superior to the earlier drug, atabrine. Atabrine was very bitter, but it was hated by the soldiers who took it because it had a lot of toxic side effects. So chloroquine becomes the drug of choice for the world malaria campaign that starts in 1955 with the World Health Organization. So this malaria research program was the largest biomedical research program at that point in history. It was even bigger than the penicillin studies that were also done here. So it was a kind of Manhattan Project of malaria research. I can't emphasize how much dogs mean to this school. So the top article, Dog Referendum Battle, is one. This was so important, this local election in Baltimore was so important that it was splashed in the headlines of the American Journal of Public Health in 1950. The school, we sent our graduate students out with the medical students to help at both the University of Maryland and Hopkins, basically went out and canvassed the citizens of Baltimore and said, please don't vote in favor of this referendum, which was intended to prevent the medical schools from using impounded dogs. So the medical schools were very, very dependent on the dogs that weren't being picked up by their owners. Within the school, it was so dog-friendly here that one professor in particular, Helen Abbey, was very well known for always bringing her Pomeranians, and that's the last one she had was named Peppy. And she and Peppy are wearing their matching Ernest Stebbins teaching medals in the photo. But being able to, we may not think of surgery as a very important part of public health here, but animal surgery was very important because it allowed us to use both tissue culture and specific organs were harvested to be used in research, and that is what Walter Ehrlich is doing in the photograph on the right. William Blaylock in the medical school, who's famous for the Blue Baby operation, practiced his operation on dogs before he did it in humans. And in this particular photo, I'm happy to tell you, he actually was operating on this Rottweiler that had been a research animal to save it after it had been hit by a car. So the surgery could also be used to help animals as well as use them for research. So sheep, rabbits, and syphilis. I've talked quite a bit about the syphilis research program here that helped to develop effective dosages for syphilis and then later all types of disease. But when we were doing our studies in the Eastern Health District, we had to test. It was an essential part of the field work to test people for syphilis. And we tested thousands and thousands of people for syphilis. And the way we did it was to use blood from sheep. And the most commonly used test was the Wassermann test. And the sheep, all they really had to do was get blood samples, and the rest of the time they could just hang out on the lawn. Because if you can imagine, at one point in history, this building was completely surrounded by green space. But things really change in the world of syphilis. One of the problems with the Wassermann test was it was not as specific as it needed to be. So there were a lot of false positives. Imagine not having syphilis and then being told, hey, you've got syphilis. Thomas Turner, who led most of the syphilis research along with Joseph Earl Moore in the medical school, had made a discovery that syphilitic rabbits produced specific antibodies for syphilis. And so he used that discovery. So two people in his lab, Robert Nelson and Manfred Mayer, in the Department of Microbiology, developed a new test in 1949 that, so some people with lupus or leprosy, the antibodies in their blood were triggering the false positives. And so the new TPI test helped to eliminate those false positives because it was so sensitive to just the antibodies for the syphilis pathogen. I think one of the most well-known stories of animals in research is our polio research done by David Bodian, Howard Howe, and Isabel Morgan in the Department of Epidemiology. And a lot of people think they must have been in microbiology or parasitology or something like that, but no, they were actually in epidemiology. But they had very extensive basic science backgrounds, and David Bodian actually had a degree in zoology, and that was absolutely essential to his polio research. So the first polio vaccines that scientists tried to develop used live virus, so it was still capable of infecting you. And what they wanted to do was give you just enough to trigger your antibodies. But what it could, and often did do, was trigger the disease. And it did that in children, and it was terrifying. So people were very, very afraid to continue to do work on a polio vaccine. But in 1947, Bodian's knowledge of monkey physiology and the very top-notch scientific skills of Bodian, Morgan, and Howe yielded the first effective live virus vaccine, which was tested in monkeys. Now, if you know about the Salk vaccine is a killed virus, and they later go with a killed virus because it is in fact safer. But Hopkins actually first experimented with a live virus vaccine in monkeys, and even when they were exposed to many times the lethal dose of poliovirus, the monkeys remained immune. So it was hailed in Time magazine as a miracle. And they used that knowledge of immunology from the monkey system to understand how polio, the pathology of polio, to both understand the progress of the disease, but also to develop effective vaccines against it. So in the 1950s, the polio research lab animals were the largest budget item in the school. It was huge. There was a whole animal facility built just for the polio research. In order to test the safety of a vaccine, the vaccine would be injected into the monkey's brain or spinal fluid, which is the system that causes paralysis when polio is present, and they would test whether it caused paralysis or other negative side effects. And the way polio research was done across the country by Jonas Salk and Bodian and everyone else was that the virus was cultured in monkey kidney cells. So monkey kidney cells were specifically well suited for growing the virus in culture. And remember, this is the early days of virology, when it was really very difficult to culture virus in vitro. So this is some of the earliest attempts to do so. So by 1955, the National Foundation for Infantile Paralysis, which is now the March of Dimes, but this is the main organization funding polio research at the time, they had purchased 30,000 monkeys over the past 15 years, and they came from India and the Philippines. And there was such a high demand for the monkeys for polio research that they actually created a monkey airlift to bring them rapidly to the United States. So what we learned in virology and pathology and epidemiology from polio, it set the stage for everything else that followed in vaccine development and evaluation. Our stature as an international institution known for its vaccine research really started in the polio research program. I interviewed this alumni, Sally Permar, not long ago, and she said this about primate research. I get this from my family. Are you killing monkeys? Is that what you do? The reason that these scientists used the monkey model is because it is one of the closest animal models to human beings. And you can do things in monkeys that you could never do with humans. So you can develop a hypothesis from your observations of humans and then test it in a monkey model to see if you're right. So this is a student of Diane Griffin's, by the way. So far we have talked about a very traditional approach to animal research that was popularized by Welch and Osler and many others in the medical and scientific communities. But the 1950s brings about several game changers. Frederik Bang and George Guy, if you've read the book, The Immortal Life of Henrietta Lacks, you know who George Guy was. But even before the book came out, many people on this faculty, I talked to Dean Clagg about George Guy, adore George Guy. He was a great, great man who made incredible contributions to science by creating a cell line that could be used for tissue culture research. I do not mean to take away from the issues raised in the Henrietta Lacks book, but that's not my main subject today. What Bang and Guy did was absolutely revolutionary. This type of culture research, which Fred Bang, he had been at the Rockefeller Institute before he came to the School of Medicine, and then in 1953, in a total surprise to everyone, they picked Bang to be chair of parasitology. And the alumni and the faculty said, well, why didn't you pick one of the famous parasitologists? Why didn't you pick Lloyd Roseboom, for heaven's sakes? Why didn't you pick Gilbert Otto, famous for his hookworm studies in dogs? What's wrong with you? I mean, people are really angry. They wrote the president of the university, they wrote the dean. Poor Fred Bang had a tough time of it in the beginning, but he was brilliant, and he totally changed the way public health research is done. So several things are going on in the 50s that are going to set up the success of his department. In addition to tissue culture research, which allowed it made research much, much more both efficient and actually cheaper and more effective than the old strictly animal models had been. So he really zeroed in on the importance of looking at the cellular level of these public health problems. Infectious disease was considered a completely different problem from cancer at this time, But Bang was one of the very early people that recognized that viruses could actually cause cancer. People thought he was just crazy. They said, you've got to be nuts. Cancer has nothing to do with infectious disease. There was also concern, beginning in the 50s, with population growth. And so the types of research that could address population-wide health problems and the fears that were growing about the impact of human population growth on the environment, and they were worried that after World War II, which was a horrible war, that the growing populations around the world would destabilize governments and cause World War III. And this population curve, it's from 1979, but it shows, you can see, no matter what the three models are, the population goes way, way up, and they predicted this would happen like in the mid-21st century. And then the population goes way, way down. They basically predicted that population growth was going to be so catastrophic that a lot of people would die. Okay, so this is what people were thinking at this time. So Frederik Bang combined elements of population dynamics, basic research, basic science laboratory research in both infectious and parasitic disease, as well as cancer. And he also established a research site in Calcutta, India in the early 60s. So he was getting international health funding. He created a monster funding machine for his department. And he did so with the great assistance of the rat man, David E. Davis. He was on the parasitology faculty from 1945 to 60, and although he didn't start the rat control program at Johns Hopkins, that was by a man named Kurt Richter in the medical school, he took it and really ran with it. And instead of focusing rather narrowly on just trying to address rats in Baltimore City as a public health problem, he saw rats as the key to the public health research kingdom. He said, you can learn so much about behavior, population dynamics, all sorts of things, in addition to just disease transmission from rats, which I think are really yucky. And our researchers have confirmed me in that. Our recent research has said that people who live around rats get much more depressed than those who don't. So I agree with them completely. So Davis built his little post-World War II rat control program into the Division of Vertebrate Ecology, first with Rockefeller Foundation funding and then with Public Health Service funding. And he was head of the division from 1950 to 1960. So if you look at the pictures up there, like, he was building empires for these rats. There were these elaborate habitats, like look at the color picture second from the left. And they were, the one on the far right is actually a map of the city block that he was tracking exactly where the rats lived and where they went to and how far was their range of habitat. And man, that man knew more about rats than just about anybody alive. And so this population biology approach to public health research, when Fred Bang came in 1953, he said, that's what I want to do. That is fantastic. Let's do that, not just with rats, but with lots of other animals. So when Bang was named chair of parasitology in 1953, as I said, he came into a lot of opposition because he wasn't strictly a parasitologist. He had done other types of medical research, but he did have experience with tropical disease when he was serving in World War II. So he wasn't ignorant of parasitology by any means, but he wasn't the titan of parasitology that we wanted to head the world's most famous department. He took a very interdisciplinary, very unorthodox, and I would say a pan-species approach to understanding what happens when disease hits a population. I looked up in the Oxford English Dictionary the term pathobiology. And although it was in use before Frederik Bang came along, it actually cited Fred Bang as initiating the use of pathobiology to mean this biology of populations approach. So he literally did coin this term. And you didn't see any medical school or veterinary school departments named pathobiology until after he started popularizing this model. So now we actually have quite a few veterinary schools with departments of pathobiology. He drew from many different disciplines, biology, ecology, zoology, but also the social sciences. He recognized, especially with help from Paul Lemkow in mental hygiene, he recognized that animal behavior was also very, very useful as a model for human behavior. With this funding that came rolling in, it's important to remember that in the 1950s, especially in the late 50s, public health started to go through this identity crisis. And people were basically making fun of public health research as not very rigorous and far behind the curve that had been established after World War II with medical and basic science research. So Bang was really reclaiming the legitimacy and the power of public health research in a number of different ways. And he built new facilities for biochemistry. He worked very closely with Roger Harriot in biochemistry, for tissue culture, and he was an expert in electron microscopy, which he had learned at the Rockefeller Institute, and he brought the first electron microscope to campus. So again, people thought he was crazy, but it was already known that there were just a few viruses that actually caused cancer in a test tube at least, and the Rouse tumor virus, which was a bird virus, was one of them. So we did a lot of early research on the Rouse tumor virus, and Kirti Shah actually was one of the people that did that. So there's this, one of the real hinge points in my book is 1957. There is like a smackdown between the very traditional basic science researchers that were headed, most of all, by Thomas Turner, chair of microbiology, and he was very much in the Welchian mold of basic science research, versus Fred Bang and Paul Lemkow and other people in the school that wanted to really take this totally unique, different approach to public health research. And some of the basic scientists just were horrified. They said, you cannot do this. This is not what William Welch would want, you know? And they said, well, the Bang contingent actually won out. The MPH science requirement, we were one of the few schools of public health to even have a required science course for the MPH. In 1957, it changed to Bang's Biology of Populations course. What did Thomas Turner do? He left the school and became Dean of Medicine and brought a lot of our basic science faculty with him. But by 1960, pathobiology had the largest budget of any department in the entire school because it was bringing in money from cancer, environmental health, behavioral sciences, and international health. It was an incredibly successful and dynamic department. The 60s were, again, a series of turning points. Abel Woolman and Cornelius Kruse, and no, I am not related to Kruse, as he was called, they were engineers, so they were coming at this from an engineering perspective. They championed the classic concept of building public health on permanent environmental alteration. How does that sound to Rachel Carson in Silent Spring? Not so good. So they were really old guard in that way. And they were angry that the traditional scope of public health research and its intimate relationship with sanitary engineering was dissolving at this point. And they saw this new type of environmental research that Bang and the public health service was propagating. They saw it as just a bunch of bunk. They didn't like it. Rachel Carson in Silent Spring, and it's important to note that she got some of her ideas about ecosystems and the effects of pesticides through studying population biology with Raymond Pearl, who is the first chair of biostatistics, but also was a very prominent biologist who was known for applying scientific and statistical principles on a population-wide basis. Okay, so that's been really kind of part of our DNA of the school since the beginning. The Public Health Service in 1964 is starting to catch the wave, and they send out these invitations for universities to apply to create institutes of environmental health studies. It's going to revolutionize the entire approach to environmental health hazards. Interdisciplinary study of biological, physiological, and social components of the rapidly changing environment. Who does that sound like? Sounds exactly like Bang. So his ideas were really getting very influential. And President Johnson gets on board and increases support for environmental research. And by 1970, we have the founding of the Environmental Protection Agency, very much because of both these population and environmental biology ideas, as well as Rachel Carson and other environmentalists who are exposing and digging into the problems of pollution and pesticides and modernization. And the idea, one of the very important ideas in all of these studies is the idea of sentinel organisms. So some of our faculty in pathobiology studied bottom-feeding birds, that would go down to the bottom of the Chesapeake Bay and scoop up their meal, because they could warn of rising pollution levels. So that gave further legitimacy and funding to the type of research that Bang was doing. So there's a major shift during the 60s from focusing on a single agent insult, so one thing that is causing, that is toxic and is causing health problems, to a much broader total cause and effect relationships approach to humans, animals, and environment. So really what's now modern environmental health. So I called this talk the Glass Menagerie because, of course, we are in this big glass building, and the Public Health Service and Fred Bang and other people are saying, yes, we want to help human beings. We want to help solve the problems of human illness. But animals and other biological systems will really help us do that. Now, I got to say, some of the folks at our school had very little to do with human health. And this, so things really accelerate in the 60s and 70s. So these are just a quick rundown and incomplete of some of the research that was being done. But this building was teeming with animals. There were all sorts of different animals. Turtles were walking around the halls for the mental hygiene comparative behavior studies of how do turtles get home. I want to know that. And they had little radio transmitters on them, so you could track that. It was so cool. It was so cool. One of the real giants in this type of research was William Sladen, who was graduated in pathobiology with a doctorate in 1957. A lot of the people doing this type of research, it's important to note, had their background or first got into research with Operation Deep Freeze and other Arctic and Antarctic research programs that were funded by the federal government beginning in the late 40s. And actually the president of Johns Hopkins at that time, Detlef Bronk, was a key national figure in promoting Antarctic research. And so Sladen banded more than 6,000 seabirds during his career, and they tracked the population growth as well as the migration patterns of these birds. So this gave them a lot of different information. It showed how the populations of birds changed, decreased or increased. It showed what kinds of diseases they suffered from. And it really was a population-wide approach to understanding the effect of disease in this particular species. He also looked at the behavior of Alaskan fur seals. In 1963, I found this great document at the Maryland History Archives. South Africa, there was like an international science exchange program going on, and at the top of the letterhead, there's like this atomic symbol thing up at the top. And the scientists in South Africa agreed to send 4 penguins to Sladen for his research. And they lived in a shopping center in East Baltimore, at East Point Shopping Center, in the Hochschild Cohn's front window. And I interviewed Elsie, anyway, one of the secretaries who worked for William Sladen and Fred Bang would go and help take care of and feed the penguins. This was an era when the Hopkins Field Station, part of the Chesapeake Bay Center, now the Chesapeake Bay Foundation, I believe, really took off at Johns Hopkins. So one of the experiments that they did to use a species as a sentinel organism was Aberdeen Proving Ground, which is north of Baltimore, had been testing military explosives for a couple of decades, and they didn't really know what the environmental impact of that was. So in the 70s, Sladen and Charles Rohde in biostatistics went slogging around through the mud in the Chesapeake, tracking baby turtles, and they gathered enormous amounts of data on the turtles. And they picked turtles because they had such a small home range, and so they were a good indicator of what was going on in that particular area. And Sladen was actually the model for a character in the movie Fly Away Home from 1996, because he was one of the national experts on North American waterfowl. So I don't know if you've seen that movie or not, but the character Dr. Killian is actually based on Dr. Sladen. Penguins! Penguins are in the school catalog in the 1970s. And if you can see this, they're actually featured in the behavioral sciences catalog entry because they were being used to study social behavior in animals. This type of research examined very complex relationships among people, pathogens, vectors, and local environments. And they did it not only in polar areas, but they also did it in tropical regions and here in the Chesapeake. So in 1961, Fred Bang and several people at the medical school get a major NIH grant to found the Center for Medical Research and Training in Calcutta. Now, our oral rehydration solution research comes out of there, but it was also a major ecological field research site. And Charles Southwick, who also studied penguins, he makes an interesting comment here. He says, I applied for Smithsonian funding so my group wouldn't feel the need to justify my ecological work in medical terms. So it speaks to the broad-based nature of this research, but you can also see why he's going to get in trouble, okay? The article at the bottom was published in Nature in 1966, so Sladen's work did have a very important finding for human health. He found that DDT residues had spread so far in the ecosystem that he actually found them in blood samples from penguins in Antarctica and seals. So that when that hit in 1966, just a few years after Silent Spring, that really triggered even more interest in funding in this type of research. I'm going to quickly go over the importance of veterinary medicine. So I've been talking about these different animal models. We have 3 DVMs on our current faculty, Megan Davis, Kathleen Gabrielson, who's also in EHS, and then Erica Duger in international health. According to my research, in the 60s and 70s, about 2.5% to 3% of public health graduates from schools of public health around the country were DVMs. So they played an important but fairly small representation in the student body. I also use the faculty CV database to do searches for a couple of different terms. Animal comes up on 177 of our faculty's CVs. Mouse, 114. So you can kind of see some of these terms that indicate just the breadth of animal research that we are still doing at the school. I'm going to skip over Harvey Fishman. He was a very prominent veterinary researcher, a veterinarian who did very important research, but I see that our time is running short. So these are some of the examples of the kind of current research. The top 3 names, they are editors on the editorial boards of these major veterinary journals. And then John Groopman's work in aflatoxins has both veterinary and agricultural significance. Ellen Silbergeld has done very important work on the presence of antibiotic resistant organisms in both farm animals and the workers and children who are exposed to them. And then finally, Joanne Zerlo, who is here today, has done very important research on issues of providing humane and adequate care for research animals. One of my very favorite types of research in the school was the seal tank. If you've ever gone on a tour of the school with me, you've heard me talk about the 5,000-gallon seal tank in the basement. People didn't believe me, but look, here's proof. We really had a seal tank. This was G. Carleton Ray, and he was featured in the Baltimore Sun several times. So the third picture from the left is him in a scuba diving suit in Antarctica, and he was the first to scuba dive to do Antarctic marine mammal research. Now, we think of seals as really, really cute, but that lower left picture that's a first seal's jaw, just to show you how incredibly strong it is, and they're like lifting up the mouth to show the teeth. So that really cool little capsule thing, looks like a spaceship, was used to do undersea research. So a lot of these faculty not only were very prominent in public health, but also did research at places like Woods Hole in Massachusetts, one of the most famous oceanographic research sites. So this is what the tank actually looked like. It was about 12 feet square, and it had portholes on the side so that you could see inside. Ray had been the curator of the New York Aquarium before he came to Johns Hopkins. And he also was quite famous for being one of the main people to spearhead passage of the Marine Mammal Protection Act, Save the Whales. So why did this go away? There are 2 reasons. Toxicology didn't have to mean the end of this really broad-ranging environmental research. In fact, it was quite compatible with it. But the development of increasingly sophisticated toxicological research methods at the molecular and cellular levels moved the school back toward an earlier, more lab-based model of science research. But in 1970, there were more than 30 faculty in the school doing environmental science research, and Bang's own department at its heyday in the mid-70s had 30 faculty in just that department. So in 1976, when the Department of Environmental Health Sciences was founded by merging environmental health, which used to be sanitary engineering, environmental medicine, where Anna Bacher was, and radiological science, that became the largest environmental health sciences department in the entire country. And it was bigger than a lot of entire schools of public health. It was huge. However, this type of animal research, having seal tanks, for heaven's sakes, in the basement, it took up a ton of space and it ate up a lot of money. And the funding streams began to dissipate. When D.A. Henderson became dean in 1977, he really viewed pathobiology as peripheral to public health. As we saw with Southwick's comment, I don't need to justify my ecology research based on human health. I just want to go do cool research. So some of these faculty could have been just as at home in other divisions of the university, but they chose to come to Johns Hopkins School of Hygiene. So Fred Bang, you can see the transformation from 1955 to 1980. It was rumored that he grew a beard to basically protest Dean Henderson. I'm not sure if that's true or not. But after he died of a heart attack in 1981, Noel Rose was appointed department chair. And he renamed, once again, the Department of Immunology and Infectious Diseases. And then after Diane Griffin takes it over, she again renamed it. So this department, what is now MMI, has gone through the most name changes of any department in the school, because each of the chairs has had a different philosophy of what they wanted to do in infectious disease research. And Noel Rose, by the way, becomes one of the world's experts on autoimmune diseases, and that's a very important area for the school even today. So the toxicology research was especially useful in focusing on pesticides and determining what are the effects of toxic levels of pesticides at the very most basic level, and why do they cause harm to human beings. So this sea change in environmental health research yields two major centers that are still with us today, and each of their directors, I'm very thankful to say, are here with us today. So in the late 70s, the animal rights activist Henry Spira had begun to picket outside of major department stores in New York City and protest the furs in the stores and say that that was inhumane and unethical to use animals for their fur in that way. He placed a full-page New York Times ad in 1980 with a rabbit with tape over his eyes, and it said, "How many rabbits does Revlon blind for beauty's sake?" Do you think that hit the cosmetic industry really, really hard? It did. So under Henderson, Henderson was really trying to promote both frontline public health practice and greater cooperation between the school and corporations and industry in both occupational health, but also as a funding stream for research at the school, as things were getting really dicey with federal funding, as they are again today. So the Center for Alternatives to Animal Testing was a really brilliant constellation of top-notch scientific tissue culture research that had been in place since Fred Bang and George Guy, but also it provided a very viable alternative to the old animal models of testing for both medical research and consumer product safety. So Ellen Silbergeld, who's also here with us today, told me in an interview, we had horrible attitudes toward the treatment of animals in the 1970s. You didn't have to justify animal use for research. You just order some animals, and there was no sense of the sanctity of their lives. That really changes throughout the 1980s, in part due to the efforts of the Center for Alternatives to Animal Testing, which succeeds in changing the guidelines of the FDA to utilize tissue culture as the preferred method of testing products. But it's also, very good science. And so the old types of tests that used as many as 22 million animals annually are dramatically brought, those numbers are dramatically brought down by the changes in FDA guidelines in the late 80s. The second center to come out of this relationship with the school and Henry Spira, the animal activists, is the Center for a Livable Future. So the picture on the top is a picture of factory farming of poultry. And Bob Lawrence, who had been at the Rockefeller Foundation and also had been a very pioneering person in the field of human rights and health, he wrote to Henry Spira and asked him for funding and help with establishing this center. And you remember I mentioned Abel Woolman and Cornelius Kruse and said they were really old school? Well, his son, Gordon Woolman, who is chair of, yes, really, Doggy, Department of Geography and Environmental Engineering, got together with Lawrence and Spira and helped to found the center in 1996. So the Center for a Livable Future has really brought many of these threads that I've been talking about together in a very powerful way. Going all the way back to William Henry Welch and his hog cholera, Bob Lawrence now writes editorials urging the hog industry in Maryland and in other places to clean up its act. And the connections among agriculture, the environment, population growth, public health, the health of communities, the fears of our resources being depleted, those are all coming together in the Center for a Livable Future. And one of its most famous advances has been the Meatless Monday campaign, which was established with the former ad executive Sid Lerner, with CLF as the advisor. So we still see the Meatless Monday campaign here at the school and around the world. So I hope I have, so this is my talk. It's clear that we really wouldn't have the school we have today, and it wouldn't be as internationally recognized for its research without the contributions of animals. These methods and priorities changed dramatically from the days of William Welch and Robert Hegner and the canaries from pet stores to the kinds of methods now being used in CLF and CAT and our science departments. And finally, from both a global and an environmental health perspective, the place of animals has been completely transformed and made a central ethical issue, which, as it should be within the ethics of public health. Thank you very much. So, questions? If you want to ask a question, can you please use this microphone? Questions?
Thanks, that was terrific. More of a comment. Plasmodium falciparum does not have an animal reservoir, but Plasmodium vivax does. There's a lot of research done at the CDC using owl monkeys to characterize the vivax malaria. And then Plasmodium gergii is a mouse Plasmodium species that also, but I gather nobody here at Hopkins was working on either of those models.
I have not run across that, no. I could have picked a different example like Lyme disease. Anyone else? Alan?
Karen, thank you very much for this great summary. For me, it was very historical because I knew, fortunately, many of those people that went through. And as you're going through it, you had this huge amount of information. I can tell you that there's more about almost each and every one of those people. But I will say one thing. I used to bring my kids to watch the seals. At the basement. It was really sort of fun. It was like going through a very special exhibit. And they would swim around for you until they said, enough of this, and then they would start splashing. So by the time you left soaking wet.
Yeah. That's great. Anyone else? Thank you very much.